All right,
God bless.
I'm doing this Harvard thing. Let's go through this fifth edition, Essential Cell Biology. This is Albert's. We're going to do an hour or so a piece in an entire Harvard undergraduate curriculum. So UCSF. Published by Norton, came out in 2019. What's up?
Bio lesson here?
Yeah, just going through an entire Harvard undergraduate coursework, at least in STEM. I'm going to probably skip some of the humanities stuff. We could do that if people want, but we'll just try STEM.
Awesome.
All right, this is the preface. Nobel Prize winning physicist Richard Feynman once noted that nature has a far, far better imagination than our own. Few things in the universe illustrate this observation better than the cell. A tiny sack of molecules capable of self-replication, this marvelous structure constitutes the fundamental building block of life. We are made of cells. Cells provide all the nutrients we consume, and the continuous activity of cells makes our planet habitable. Let me zoom in a little. Try to gauge how much I can see. All right. To understand ourselves and the world of which we are a part, we need to know something of the life of cells. Armed with such knowledge, we as citizens and stewards of the global community, speak for yourself, will be better equipped to make well-informed decisions about increasingly sophisticated issues from climate change and food security to biomedical technologies and emerging epidemics. In Essential Cell Biology, we introduce readers to the fundamentals of cell biology. The 5th edition introduces powerful new techniques that allow us to examine cells and their components with unprecedented precision, such as super-resolution fluorescence microscopy and cryo-electron microscopy, as well as the latest methods for DNA sequencing and gene editing. We discuss new thinking about how cells organize and encourage the chemical reactions that make life possible, and we review recent insights into human origins and genetics. With each edition of ECB, Essential Cell Biology, its authors re-experience the joy of learning something new and surprising about cells. We're also reminded of how much we still don't know. Many of the fascinating questions, the most fascinating questions in cell biology remain unanswered. How did cells arise on the early Earth, multiplying and diversifying through billions of years of evolution to fill every possible niche, from steaming vents in the ocean floor to frozen mountaintops, and, in doing so, transform our planet's entire environment? How is it possible for billions of cells to seamlessly cooperate and form large multicellular organisms like ourselves? These are among the many challenges that remain for the next generation of cell biologists, some of whom will begin a wonderful lifelong journey with this textbook. Readers interested in learning how scientific inquisitiveness can fuel breakthroughs in our understanding of cell biology will enjoy the stories of discovery presented in each chapter's How We Know feature. Packed with experimental data and design, these narratives illustrate how biologists tackle important questions and how experimental results shape future ideas. In this edition, a new How We Know recounts the discoveries that first reveal how cells transform the energy locked in food molecules into the forms used to power the metabolic reactions on which life depends. As in previous editions, the questions in the margins at the end of the chapter not only test comprehension but also encourage careful thought and the application of newly acquired information to a broader biological context. Some of these questions have more than one valid answer, and others invite speculation. Answers to all the questions are included at the back of the book, and many provide additional information or alternative perspective on material presented in the main text. More than 160 video clips, animations, atomic structures, and high-resolution micrographs complement the book and are available online. The movies are correlated with each chapter, and callouts are highlighted in color. This supplemental material, let's get my teaching assistant here. This supplemental material, created to clarify complex and critical concepts, highlights the intrinsic beauty of living cells. For those who wish to probe even more deeply, Molecular Biology of the Cell, now in its 6th edition, offers a detailed account of the life of the cell. In addition, Molecular Biology of the Cell, 6th edition, A Problems Approach by Wilson and Hunt, provides a goldmine of thought-provoking questions. at all levels of difficulty. We have drawn upon this tour de force of experimental reasoning for some of the questions in essential cell biology, and we are very grateful to its authors. Every chapter of ECB is the product of a communal effort. Both text and figures are revised and refined as drafts circulated from one author to another, many times over and back again. The numerous other individuals who have helped bring this project to fruition are credited in the acknowledgments that follow. Despite our best efforts, it is inevitable that errors have crept into the book, and we encourage eagle-eye readers who find mistakes to let us know so that we can correct them in the next printing. Here are all the acknowledgments, and we will move on to Chapter 1. Let's look at the authors very briefly. Bruce Alberts received his PhD from Harvard and is professor in the Department of Biochemistry and Biophysics at UCSF, editor-in-chief of Science, wow, from 2008 to 2013. That's one of my favorite journals. And served as the president of the US NAS from 1993 to 2005. Wow, very cool. I know. Yes, we're going to talk about that too. Yes, we're doing linear algebra next. Shut up, kitty. Wait your turn. I know you already know biology. Yeah. Well, if you could teach it better, come on up here and do it. Psh. Shut up. That's not even a real meow. You don't even know how to meow. Come here. She just wants to play. She wants attention. Let's play. You want to play? Let's play for 1 minute. We'll take a 1 minute break to study the biology of the wild beast cat. Yeah? Yeah? What? Yeah. You want a treat? Yeah. I'll get you a treat. Hold on. Let's get you some treats. You want some treats? Fine. F you too. All right, she's licking her mouth. That means she's hungry. She wants some treats. There we go. Now you'll be quiet. All right. Chapter 1. Cells, the fundamental units of life. It's true. What does it mean to be living? Petunias, people, and pond scum are all alive. Stones, sand, and summer breezes are not. But what are the fundamental properties that characterize living things and distinguish them from non-living matter? The answer hinges on a basic fact that is taken for granted now, but marked a revolution in thinking when first established more than 175 years ago. All living things or organisms are built from cells, small, membrane-enclosed units filled with a concentrated aqueous solution of chemicals and endowed with the extraordinary ability to create copies of themselves by growing and then dividing in two. The simplest forms of life are solitary cells. Higher organisms, including ourselves, are communities of cells derived by growth and division from a single founder cell. Every animal or plant is a vast colony of individual cells, each of which performs a specialized function that is integrated by intricate systems of cell-to-cell communication. Just so you know, we're going to talk about unity and diversity of cells, cells under the microscope, the prokaryotic cell, the eukaryotic cell, and model organisms. Cells, therefore, are the fundamental units of life. Thus, it is to cell biology, the study of cells and their structure, function, and behavior, that we look for an answer to the question of what life is and how it works. With a deeper understanding of cells, we can begin to tackle the grand historical problems of life on Earth, its mysterious origins, its stunning diversity produced by billions of years of evolution, and its invasion of every conceivable habitat on the planet. At the same time, cell biology can provide us with answers to the questions we have about ourselves. Where did we come from? How do we develop from a single fertilized egg cell? How is each of us similar to, yet different from, everyone else on Earth? Why do we get sick, grow old, and die? In this chapter, we introduce the concept of cells, what they are, where they come from, and how we have learned so much about them. We begin by looking at the great variety of forms that cells can adopt, and we take a preliminary glimpse at the cellular machinery or chemical machinery that all cells have in common. We then consider how cells are made visible under the microscope, and what we see when we peer inside them. Finally, we discuss how we can exploit the similarities of living things to achieve a coherent understanding of all forms of life on Earth, from the tiniest bacterium to the mightiest oak and the mightiest cat, right? If you're just joining us, we're doing 5 different Harvard courses this morning. And we have a very loud guest lecturer who wants to be heard every moment. I'm sorry about that. A big part of my live streams before I took a 4 or 5 year vacation was education. And I thought it'd be fun to download 5 Harvard textbooks that your average Harvard student would be studying and read those textbooks. And some people may want to listen in along. Some people may have learned this a long time ago. Some people may not give a crap and just are here for the cat or my beautiful face. But regardless, that's the program for today. Biologists, so let's talk about the unity and diversity of cells. Biologists estimate that there may be up to 100 million distinct species of living things on our planet. Organisms as different as a dolphin and a rose, or a bacterium and a butterfly. Cells, too, differ vastly in form and function. Animal cells differ from those in a plant, and even cells within a single multicellular organism can differ wildly in appearance and activity. Yet, despite these differences, all cells share a fundamental chemistry and other common features. In this section, we take stock of some of the similarities and differences among cells, and we discuss how all present-day cells appear to have evolved from a common ancestor. Yeah? Cells vary enormously in appearance and function. When comparing one cell to another, one of the most obvious places to start with is size. A bacterial cell, say a lactobacillus, in a piece of cheese, is a few micrometers, or micro-m. This is the Greek letter mu. If you haven't seen it before, I have my Wacom tablet. Maybe I should plug it in. I don't know where the stylus is, though. The stylus would probably help, wouldn't it? I'll have to find that. Shut up, kitty. Yeah, I think this isn't too useful without the stylus. Well, anyway, I was going to just sort of point out, we all know what 1 meter is, right? How long is a meter? It's around 3 feet. 3 feet, right? It's like 3.2 feet exactly, I think. So it's almost like a yard. Let's see. 1 meter, 2 feet. 3.28 actually. So it's more like 3.3. 3.28084. All right, so 1 millimeter is 1,000th of that. So we know how big a millimeter is, right? It's very, very thin. Extremely small distance. Centimeter is about the length of a finger knuckle. So a millimeter is 1/10 of a centimeter. So a micrometer is 1/1000th of a millimeter, and 1/1,000,000th of a meter. Is a micrometer visible? This is supposed to be micrometer. No, it's not visible. It's
I wouldn't think it would be.
just below the threshold of, probably we lose the ability to discern maybe around like 100 micrometers. So 1 micrometer is too small. So that's about 25 times smaller than the width of a human hair. So that's the diameter maybe, or length of a bacterial cell. So not quite so microscopic. It's microscopic, but it's not like it's so small it's almost, impossible to see. It's definitely not within the ability for us to discern, but it's not like it's that far away. It's not like it's an atom or something. At the other extreme, a frog egg, which is also a single cell, has a diameter of about 1 millimeter, which is technically visible. If we scale them up to make the lactobacillus the size of a person, the frog egg would be half a mile high. Cells vary just as widely in their shape. A typical nerve cell in your brain, for example, is enormously extended. It sends out its electrical signals along a single fine protrusion called an axon that is 10,000 times longer than it is thick. So it's very long, like a piece of string almost. And the cell receives signals from other nerve cells through a collection of shorter extensions that sprout from its body like the branches of a tree. A pond-dwelling paramecium, on the other hand, is shaped like a submarine and is covered with thousands of cilia, hair-like projections whose sinuous, coordinated beating sweeps the cell forward, rotating as it goes. A cell in the surface layer of a plant is squat and immobile, surrounded by a rigid box of cellulose with an outer, waterproof covering of wax. A macrophage in the body of an animal, by contrast, crawls through tissues, constantly pouring itself into new shapes as it searches for it and engulfs debris, foreign microorganisms, and dead or dying cells. A fission yeast is shaped like a rod, whereas a budding yeast is delightfully spherical, and so on. Cells are enormously diverse in their chemical requirements. Some require oxygen to live, for others the gas is deadly. Some cells consume little more than carbon dioxide, sunlight, and water as their raw materials. Others need a complex mixture of molecules produced by other cells. These differences in size, shape, and chemical requirements often reflect differences in cell function. Some cells are specialized factories for the production of particular substances such as hormones, starch, fat, latex, or pigments. Others, like muscle cells, are engines that burn fuel to do mechanical work. Still others are electricity generators, like the modified muscle cells in the electric eel. I don't know if I'd go to Harvard to do a postgrad, but some modifications specialize the cell so much that the cell ceases to proliferate, thus producing no descendants. Such specialization would be senseless for a cell that lived a solitary life. In a multicellular organism, however, there is a division of labor among cells, allowing some cells to become specialized to an extreme degree for particular tasks, and leaving them dependent on their fellow cells for many basic requirements. Even the most basic need of all, that of passing on the genetic instructions of the organism to the next generation, is delegated to specialists, the egg and the sperm. Living cells have a similar basic chemistry. Despite the extraordinary diversity of plants and animals, people have recognized from time immemorial that these organisms have something in common, something that entitles them all to be called living things. But while it seemed easy enough to recognize life, it was remarkably difficult to say in what sense all living things were alike. Textbooks had to settle for defining life in abstract general terms related to growth, reproduction, and an ability to actively alter their behavior in response to the environment. Let's actually look here at question 1.1. Life is easy to recognize but difficult to define. According to one popular biology text, living things are 1, highly organized compared to natural inanimate objects. 2, they display homeostasis, maintaining a relatively constant internal environment. 3, they reproduce themselves. 4, they grow and develop from simple beginnings. 5, they take energy and matter from the environment and transform it. 6, they respond to stimuli. 7, they show adaptation to their environment. Score of person, vacuum cleaner, and a potato with respect to these characteristics. Okay. The discoveries of biochemists and molecular biologists have provided an elegant solution to this awkward situation. Although the cells of all living things are enormously varied when viewed from the outside, they are fundamentally similar inside. We now know that cells resemble one another to an astonishing degree in the details of their chemistry. They are composed of the same sorts of molecules, which participate in the same types of chemical reactions, discussed in Chapter 2. In all organisms, genetic information, in the form of genes, is carried in DNA molecules. This information is written in the same chemical code, constructed out of the same chemical building blocks, interpreted by essentially the same chemical machinery, and replicated in the same way when a cell or organism reproduces. Let's take a break to look at figure 1.1. So we're looking at 100 micrometer resolution here. Figure 1.1, cells come in a variety of shapes and sizes. Note the very different scales in these micrographs. This one's 25, 25, 5, and 3. Drawing of a single nerve cell from a mammalian brain. This is here. A. This cell has a single unbranched extension, the axon. You can see it there. Projecting towards the top of the image, though through which it sends electrical signals to other nerve cells, and possesses a huge branching tree of projections, these are called dendrites, through which it receives signals from as many as 100,000 other nerve cells. And again, notice the scale here. This is 100 micrometers, almost visible amount, or length, I should say. This is a paramecium, a commonly studied protozoan. A single giant cell swims by means of the beating cilia that covers its surface. The surface of a snapdragon flower petal displays an orderly arrangement of packed cells. D is a macrophage, spreading itself out as it patrols animal tissues in search of invading organisms. And a fission yeast, is also caught in the act of dividing into two. The medial septum, stained red, is forming a wall between the two nuclei, also stained red, that have separated into two daughter cells. The cell membranes are stained green. All right. In every cell, long polymer chains of DNA are made from the same set of 4 monomers called nucleotides. Very important, nucleotides. This is where we get mRNA, which we'll talk about in a second. Strung together in different sequences, like the letters of an alphabet. The information encoded in these DNA molecules is read out, or transcribed, very important, transcribed, into a related set of polynucleotides called RNA. Although some of these RNA molecules have their own regulatory, structural, or chemical activities, most are translated. So we have transcribed and translated. So DNA gets transcribed into RNA, and RNA gets translated into a different type of polymer called a protein. So a polymer is just any molecule with repeating units. Any molecule with repeating units. That includes plastics or polymers, but also biomolecular molecules too. This flow of information from RNA to DNA, from DNA to RNA to protein is so fundamental to life that it is referred to as the central dogma. The central dogma of biology. And here's a look at it from this side. DNA, RNA, protein. DNA synthesis replicates. That's not too important right now. That's when cells divide and things like that. But RNA synthesis is when we go from DNA to RNA. These are both nucleotides. There's some subtle differences that make RNA, RNA, and DNA, DNA. But either way, from this RNA template, we can translate this into proteins, and these proteins are made of amino acids. So that's the central dogma. The appearance and behavior of a cell are dictated largely by its protein molecules, which serve as structural supports, chemical catalysts, molecular motors, and much more. Proteins are built from amino acids. There's one thing you need to know in biology, this is it. And all organisms use the same set of 20 amino acids. We have 26 letters in English, but God has 20 amino acids to make their proteins. But the amino acids are linked in different sequences. Just the same way we can make a lot of different words with 20 amino acids, we can make a lot of different proteins
with, we can make a lot of words with 26 alphabet letters, excuse me. We can make a lot of different proteins with 20 amino acids. But the amino acids are linked in different sequences, giving each type of protein molecule a different three-dimensional shape or conformation. So even the change of one amino acid in the wrong place can create a disastrous situation. Let me show you. We're going to look up some protein sequences using this tool from the government. Thank you, government. Well, let's do a different. Let's go here. So this is dystrophin, which is the gene for the protein that, when mutated, causes muscular dystrophy. You can see it's in all different types of organisms. So I'm just going to type in dystrophin homo to get Homo sapiens. And if I look at this gene and I want to know the sequence, I can click to get the sequence. I haven't used this website in years. It should be here somewhere. Here it is. So this is the DMD. This is a gene that's on the X chromosome. That's why it's an X-linked disorder. So only boys get muscular dystrophy or Duchenne muscular dystrophy because girls have two copies of the gene. They have an X copy and a Y copy, or an X copy and an X copy. Boys only have an X copy, one copy, because they have a Y chromosome. So if you mutate the one copy, there's no backup copy. Females have a backup copy. So here's the gene sequence, and you can see it's a very intricate detail, but one, and it goes on and on. In fact, dystrophin is one of the biggest genes in the human body. One misspelling of this, just one of these, right? How many are there? What does it look like? Look at the bottom of my screen. You can see the scrolling is going up very slowly. What would you guess, 1 million? It's enormously large, right?
Even
2 million. So there's all kinds of tools in here to go through it, but it's an enormous protein. And there's all kinds of different ways you can look at these. And again, if you want to do this yourself, just go to the NCBI. But yeah, I think that you can get a sense for just, this is a very large protein, or gene I should say, but there are many genes that are smaller. There's most genes, in fact, are a lot smaller than this one. But you can see that this gene that encodes for this important protein called dystrophin, it's such an important protein when it comes to muscle that a single wrong nucleotide can cause death, unfortunately, in people. So let's keep going. All right, so amino acids are linked in different sequences, giving each type of protein molecule a different three-dimensional shape or conformation, just as different sequences of letters spell different words. In this way, the same basic biochemical machinery has served to generate the whole gamut of life on Earth. Right, in all living cells, genetic information flows from DNA to RNA and from RNA to protein. That's transcription from DNA to RNA and translation from RNA to protein, an arrangement known as the central dogma. The sequence of nucleotides in a particular segment of DNA, which is a gene, is transcribed into an RNA molecule, which can then be translated into the linear sequence of amino acids of a protein. Only a small part of the gene, the RNA and protein, is shown. All right, living cells are self-replicating collections of catalysts. That's a great way to put it. One of the most commonly cited properties of living things is their ability to reproduce. For cells, the process involves duplicating their genetic material and other components and then dividing into two, producing a pair of daughter cells that are themselves capable of undergoing the same cycle of replication, sort of an exponential growth. It is the special relationship between DNA, RNA, and proteins, as outlined in the central dogma, that make this self-replication possible. DNA encodes information that ultimately directs the assembly of proteins. The sequences of nucleotides in a molecule of DNA dictates the sequence of amino acids in a protein. All right, proteins in turn catalyze the replication of DNA and the transcription of RNA. And they participate in the translation of RNA into proteins. This feedback loop between proteins and polynucleotides underlies the self-reproducing behavior of living things. So this is interesting, and there's one author named Douglas Hofstadter, I think, who wrote a book called Gödel, Escher, Bach, which is a very influential book. I recommend the book. It's good, not great. And anyway, the point that he makes is, and many people have observed this, is that when you look at, hold on, my cat messed up a lot of my infrastructure here. When you look at DNA, it's self-referential. What does that mean? This central dogma is called dogma for a reason. Dogma is usually used like kind of an insulting word, right? Well, the reason this is kind of interesting is that this is the central dogma. But the reality is a bit different. There's a lot of other interactions that are occurring. I won't talk too much about those just yet. That's what this whole book is about. But what I want you to understand is that, sorry, I don't know what my computer's doing right now. What I want you to understand is basically this idea that when some of these proteins, right, are related to helping DNA replicate and maintaining their status. So there's this feedback loop that's very interesting, right? There are proteins that help DNA replicate. And those proteins are created by the very DNA. So some people call this recursive in computer science. There's a lot of concepts like this in art and music, which is why it's called Gödel, Escher, Bach. And Gödel's incompleteness theorem has a recursive idea in it. So and Hofstadter's general theme of the book is that this is where like all complexity and intelligence and beauty lies in this self-referencing sort of strange loop is what he calls them. He even has something called I Am a Strange Loop. I think it's a book, and maybe even became a musical, I don't know. But anyway, it's an interesting book. But the point is, you really can't have DNA without the proteins that allow the DNA to have its elegant song and dance of this is some DNA, of basically chaperoning it to divide and separate and then rewind again and come apart and then having the cells kind of divide. This is two cells dividing. Starts off as one cell, and you can see them kind of budding out, and then they separate. Proteins are required to do this, but wait a second, the data for the proteins are stored in the DNA. The RNA is kind of like this intermediate. So, it's sort of like this very funny thing. Well, if DNA comes from proteins and proteins come from DNA, how does this all work? And it's kind of amazing to think about. And I think the thing about Hofstadter, though, is a good scientist and everything, but the fact that this like blew his mind is probably a little like memey, and it kind of reminds me of like maybe the stereotypical pothead or something. Saying, "Whoa, bro, this is crazy." The reality is it's very explainable and not that deep as maybe the connections that he's drawing that he thinks they are. So anyway, that's just my 2 cents. So let's keep going here. DNA encodes information that ultimately directs the assembly of proteins. The sequences of nucleotides in a molecule of DNA dictates the sequence of amino acids in a protein. Proteins, in turn, catalyze the replication of DNA and the transcription of RNA, and they participate in the translation of RNA to proteins. This feedback loop between proteins and polynucleotides underlies the self-reproducing behavior of living things. We discuss some complex interdependence between these three actors in detail in chapters 5 through 8. In addition to their roles in polynucleotide and protein synthesis, remember, polynucleotide just means DNA or RNA, a lot of nucleotides. Proteins also catalyze the many other chemical reactions that keep the self-replicating system shown in 1.4 running. So let's see what 1.3 is here. These are all living organisms are constructed from cells. This is a colony of bacteria, again, 2 micrometers, not quite visible, and bacteria could be good or bad. A butterfly, a rose, and a dolphin are all made of cells that have a fundamentally similar chemistry, which is really amazing that the DNA, if you compared the DNA amongst these 4 organisms, they're very, 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 very similar. Which surprised me when I learned that the DNA contents of a butterfly and a dolphin and a rose, I mean, you can imagine these things are so different. Most of the machinery is the same. Most of the way that we harvest energy and things like that has not changed in a billion years, which is really kind of amazing if you think about it. Or maybe 500 million years, better way to look at it, or 100 million years. All right. A living cell can break down nutrients and use the products to make both the building blocks needed to produce polynucleotides, DNA and RNA, proteins, and other cell constituents. What's in a cell? We'll find out soon. And to generate the energy needed to power these biosynthetic processes. So it's using biology to make things. We discussed these vital metabolic reactions in detail in chapter 3 and 13. This word metabolic confuses a lot of people, but basically metabolic is just any kind of biosynthetic process. And we sometimes say metabolism. Some people say, well, I have a fast metabolism or slow metabolism, and I think that's, to me, is often in their head. But the point is, any transformational reaction from one entity to another can be thought of as a metabolic reaction. Let's look at figure 1.4. Life is an autocatalytic process. So auto means automatic or will occur on its own. Catalytic means something happening or something being created or transformed. DNA and RNA provide the sequence information, the green arrows, that is used to produce proteins and to copy themselves. Proteins, in turn, provide the catalytic activity needed to synthesize DNA, RNA, and themselves. So it's amazing that the proteins are kind of machines that make all this happen, and the DNA is the information. It's almost like the software and hardware to some extent. Together, these feedback loops create the self-replicating system that endows living cells with their ability to reproduce. And as you study this, I think you'll find it to be rather amazing how you can have a protein that's sort of guiding or even dozens of proteins or hundreds of proteins that are guiding and shaping the DNA and RNA so that the DNA and RNA can transform into more protein. So the very proteins that are guiding the DNA and RNA are sequenced from DNA and RNA. So there's no top level, if you will. It all starts sort of at DNA, I suppose, but there's still kind of this very intricate meta transformation. You don't usually see that in things like computer programming, where you sort of have a core level of DNA, and typically, I suppose well, maybe it is somewhat similar. I don't know. It's something very interesting to ponder and think about. Only living cells can perform these astonishing feats of self-replication. Viruses also contain information in the form of DNA and RNA, but they do not have the ability to reproduce by themselves. Instead, they parasitize the reproductive machinery, or they take over the reproductive machinery of cells that they invade to make copies of themselves. This is like the COVID virus will take over some of your cells to make a copy of itself. Thus, viruses are not truly considered living. They're merely chemical zombies, inert and inactive outside of their host cells, but able to exert a malign or malignant control once they gain entry. We review viruses in chapter 9. yeah, if you look at my DNA, there's loyalty instead of A, C, T, and G, but that's another topic. Scientists are very confused by that. Thank you, Kendrick Lamar. All living cells have apparently evolved from the same ancestral cell. This is a very, very unusual idea. All living cells have evolved from the same ancestral cell. When a cell replicates its DNA in preparation for cell division, the copying is not always perfect. I think this is probably thing number 2 to really understand in biology. This will help you understand a lot. When a cell replicates its DNA in preparation for cell division, the copying is not always perfect. Well, why? Well, we just looked at the NCBI, right? NCBI, National Center for Biotechnology Information. We can go to the genome domain or gene domain. We can type in, I don't know, insulin. Do you want to type in insulin? Any other protein some people care about? There's insulin, there's the insulin receptor, there's the insulin precursor. I'm going to type in insulin homo, again for the species, but I could do mus musculus, which is just the mouse, and we could see the insulin gene in mice. We can look at KRAS, which is a very unusual protein. Insulin is most definitely a protein. It's made from an insulin precursor. So let's look at, and then it has a receptor, and there's IGF, which is not insulin, but related. Here's an insulin-degrading protein. You can see there's dozens of proteins related to insulin, but the big one is called pre-pro-insulin, I think, or pre-insulin. I think it might be. But a lot of hormones have a pre-pro kind of concept within different, let's see. There's pre-insulin, pro-insulin, there's pre-pro in the elephant shark. Well, this is plain old insulin on chromosome 10. So maybe insulin doesn't get chopped up and metabolized. Anyway, let's go to the genome sequence. Here's some of the ways to look at the sequence, but I'm just going to go to the basic FASTA, which is just the letters, because I think that's better. There it is. It's really small relative to dystrophin, right? Dystrophin was like millions of base pairs. These are each called base pairs. And you can see there's 4 letters, A, C, T, and G. So anyway, insulin is really, really short. And we're going to learn in different chapters that these three nucleotides each code for a different amino acid. And this is around 44 amino acids, if I'm not mistaken. So we're going to determine and learn a bit about this down the road. Why did I mention this? Because when we looked at dystrophin, which was so large, it made sense to me, at least, that it was possible that you couldn't necessarily replicate this too easily, right? That maybe something would go wrong. It was millions of base pairs. I can't even scroll down fast enough to get to the bottom. This is maybe the fastest I can scroll, and it would still take me about a minute to scroll down to the bottom. So how could it be possible that this gets copied perfectly every single time? it would have to be a really good copy machine, right? We have this copy machine inside of our body that is this good. And when we think about computers, we think about copying and pasting. When you copy and paste something, it's not like one thing in the copy-paste will go wrong, right? It's not like one letter will accidentally get changed. Well, that's because our computer software is a little bit different from our body software. In our body software, every now and then, one of these will, instead of an A, T, C, or G, it might go to an A, C, T, or G in a different order. And that's all it takes for there to be a so-called mutation. And you can see just how long this was, right? This is just so enormous. How do you get it exactly the same every time? That's sometimes called fidelity. And we use that term in music or audio sometimes, hi-fi, lo-fi. So high fidelity is a perfect copy. Well, on occasion, the instructions are corrupted by mutations that change the sequences of nucleotides in the DNA. For this reason, daughter cells are not necessarily exact replicas of their parents. Mutations can create offspring that are changed for the worse, in that they are less able to survive or reproduce, or changed for the better, in that they will be better able to survive or reproduce, or changed in a neutral way, in that they are genetically different but equally viable. The struggle for survival eliminates the first, favors the second, and tolerates the third. The genes of the next generation will be the genes of the survivors. That's probably the next most important thing to understand in biology. Central dogma: cell replication does not result in a perfect copy, and the genes that survive are the genes of the survivors. Now, we notice that there are still genetic diseases despite this, right? And that's because the window for reproduction and survival are two different things. You can have Duchenne muscular dystrophy, which has a lifespan of around 20 years, and still reproduce. You can have a lifespan of 40 years with Huntington's disease and still reproduce. So it's not a perfect sort of one-to-one ratio. If you have a fatal disease that results in death before the age of 5, then the genes of the next generation will be the genes of the survivors and not the genes of that patient, for instance. So anyway, I'd encourage you to think about this a little bit, that it seems to be that most mutations are actually ones that change you for the worse. Having said that, the only ones that really carry on over long periods of time are the ones that change you for the better. So it's a very interesting sort of concept. For many organisms, the pattern of heredity may be complicated by sexual reproduction, in which two cells of the same species fuse, pooling their DNA. The genetic cards are then shuffled, redealt, and distributed in new combinations to the next generation, to be tested again for their ability to promote survival and reproduction. One way to look about or think about DNA is basically DNA is just a game. And the game is, can you reproduce and send your DNA? Here's some DNA here. Here's some DNA here. And the game is, can this DNA reproduce better or more than this DNA? And that's all there is to life, is who can reproduce the most. And if you think about it that way, it's a little bit of a cold world, but it is definitely the kind of dynamic that is sort of what gives rise to life. This DNA is competing for resources. The machines that it makes, the DNA makes proteins. So those proteins are molecular machines. It's trying to make the best molecular machines that will reproduce itself. If it can't do that, it'll die. So this DNA might make a protein organism of a cat. Well, this one made a cat, but a cat with no claws. Well, this one's going to survive and reproduce, more likely at least, than this one. And maybe that's why cats have very sharp claws. And so forth and so forth. And all the way up to perhaps even consciousness. And there's a lot of debate as to whether consciousness, what we seem to exhibit, is a byproduct of evolution or a direct cause of evolution. And we don't know. But it's a good question. All right. These simple principles of genetic change and selection, applied repeatedly over billions of cell generations, are the basis of evolution, the process by which living species become gradually modified and adapted to their environment in more and more sophisticated ways. Evolution offers a startling but compelling explanation of why present-day cells are so similar in their fundamentals. This is probably the best proof. There's a lot of proof of evolution, and a lot of it is just common sense. But one very simple proof is that if you look at the DNA of early organisms, you can actually see and chart on a tree of life, or a more complex chart, exactly when certain things happened. For example, one protein I've studied very closely is called oxytocin. Oxytocin is a hormone, sometimes called the love hormone, jokingly. It's an octamer, I believe, or nonomer. It started off as an octamer, and it actually evolved from a different hormone called vasopressin. So fish had vasopressin before humans existed, and oxytocin was made just in a molecular accident by mutating by that low-fidelity copying mechanism. Oxytocin just happened to be created. And over an evolutionary tree branch, you can see exactly when oxytocin showed up in evolution. So you can see when it showed up in organisms. And oxytocin is a fascinating little protein, sometimes called, like I said, the love hormone. You make oxytocin when you hug somebody or kiss somebody. Dogs make a stream of oxytocin when they see their owners. I don't like to use the word owner, but when they see their living companions. So it's very interesting how this protein works, but what's more interesting to me is that you can see what species have oxytocin and what species don't. Most unicellular organisms, like bacteria, don't have oxytocin. But what point did organisms grow enough to require it? And you can basically see that there's no doubt as to how this all came about. It came about by accidental replication of some of these proteins. There's no doubt whatsoever that this was a happy accident, and that humans are the production of lots of these little happy accidents. And over long enough periods of time, we can't think and conceptualize and contextualize just how long we're thinking about. We're thinking about millions and millions of years. Over enough generations, you'll see those differences. Mendel, who was a friar or a priest or something like that, right? Mendel, used pea pods, and he could see the evolution, or at least the hereditary traits, very quickly. It wasn't long before he realized, well, if you mate these two plants, this is what you get. And it isn't a surprise that over time you could influence the sort of drift of genes and species of these pea pods, because it was very quick it didn't take long to reproduce these. It takes a long time for humans to reproduce. So you don't see the changes immediately. But you could even think in your own head, if only these types of people reproduced, this would be the only progeny left, and then every now and then you'd have an accidental mutation that maybe would change hair color or skin color, and then you'd have to start all over again. So, it's just something to think about in terms of, it's pretty easy to understand from a common sense perspective. So anytime I hear somebody ask about the whether evolution is true or not, it's very frustrating to me, because it's just something born of ignorance. All proteins evolved over time. That's correct. Gemini in chat is asking. That's exactly right. Proteins evolved just like species did. Very, very interesting way to look at it. All right. So repeated application of these billions of cell generations are the basis of evolution, the process by which living species became gradually modified and adapted to their environment in more and more sophisticated ways. Evolution offers a startling but compelling explanation of why present-day cells are so similar in their fundamentals. Our cells are the same as bird cells, and those are the same as bacterial cells, or they have a lot of the same characteristics in DNA. They all inherited their genetic instructions from the same common ancestral cell. It is estimated that this cell existed between 3.5 and 3.8 billion years ago, and we must suppose that it contained a prototype of the universal machinery of all life on Earth today. And again, that's debated quite a bit. It's not 100% clear how that happened or what it looked like. And we'll probably talk about that in a minute. But it is very interesting. And perhaps there are parallel, ancestors that created life. It's possible a lot of different things happen. So we don't have all the answers there quite yet. Through a very long process of mutation and natural selection, the descendants of this ancestral cell have gradually diverged to fill every habitat on Earth with organisms that exploit the potential of the machinery in a seemingly endless variety of ways. Well, I would dispute that a little bit. We actually have quite a similar need for things like ATP and other molecular nutrients. So I'd say that we're actually more similar with other organisms than maybe it's being said here. But regardless, mutations are mistakes in the DNA that change the genetic plan from that of the previous generation. Imagine a shoe factory. Would you expect mistakes in copying the shoe design to lead to improvements in the shoes produced? Explain your answer. Unintentional changes in copying shoe design. Well, I'd say most of those changes would not be good, right? But every now and then you might say, okay, let's use rubber instead. Let's use this or that, and maybe you would make a positive change, and then you'd say, okay, let's put that in our new factory plan. All right, genes provide instructions for the form, function, and behavior of cells and organisms. And we're going to probably wrap this up relatively soon and go to the next Harvard course. We're doing 5 different Harvard courses, undergrad courses today. Thanks for joining me. It's going to be like a bit of a marathon stream. And I'm feeling very energetic today, so I'm going to try to, do a marathon as much as I can. But who knows, I might tap out at some point.